Hi everyone, I'm really happy to be here, and I have to thank the organizers as well for putting all this together, and it's really so much interesting data. And I'm here to talk about some of the sort of rather confusing data, at least I think, that we've generated over the last years. And as Ian pointed out, I'm a PhD student, or I am a PhD student, maybe, yeah, in Jan Bryson's group at Karolinska Institutet in Stockholm. And we have a slightly different angle to this maybe than some of the others, because the group is mainly, it's sort of interested in this process that we're looking at right here, which is really the cytotoxicity process of these cells. So the killing event that the NK cells and the cytotoxic lymphocytes are doing. So that's sort of the frame for the group's work. And we're working with a lot of different immunodeficiency syndromes mainly to try to decipher this process and to understand its relevance in different diseases. So first I would just like to say something short about cytotoxic lymphocytes, which we denote this together. So because we talk a lot about the NK cells, but I would just like to point out that the NK cells, they are called natural killer cells because they, when we're all born, we have a lot of different receptors that these NK cells express that can sense pathological signals both from malignant cells and from viral infected or stressed cells. And they can then, when they see a pathological pattern, they go in and kill the cell. The T cells, on the other side, they're adaptive. So when we during life experience different dangers, okay, it's moving by itself. That's very interesting. So instead, they are responding to dangerous signals that we've seen. So they're sort of they're contributing to our immunological memory, whereas these are sort of the species memory, more on a global scale. But they share very much of their effector function. So their killing is very similar, and they're also important in the same processes, namely like control of viral infection and malignancy, and also immune activation, which might be very relevant here. So just something short about like how they're activated. So they're going around the body looking for pathological cells. When they find one, they attach to them, and then they create like a synapse, pretty similar to in the neurological system, through which they release these cytotoxic proteins that then lead to the killing of the cell. And then in this process, they also release a lot of cytokines. So they're also important pro-inflammatory cytokine producers. But now, just focusing a little bit more on this process, as you see, you sort of need two things to be able to kill. What we're seeing here in the top is an NK cell or a T cell, and on the bottom here we're seeing a target cell that's being killed. So you have to be able to have proteins, perforins and granzymes, as have been pointed out before. They have to be there and be functional, and you also have to be able to release them, because some patients in specific disorders, not ME that we're talking about, but other disorders, have a dysfunction either in these proteins or in the granules, which renders them completely unable to do this. And what happens then is that you, instead of killing off the cell, you just recognize that this target, the violet cell here, that it's sick, and you produce lots of cytokines. And this leads to sort of an immune frustration situation where the whole immune system tries to do something about these sick cells, but they can't, because they have to kill them and they can't. And then what you would normally do is that you would go in and you would kill all the immune cells that are overactivated. But as you can't do that because you don't have the killing ability in the first place, the whole immune system spirals out of control. And this syndrome that I'm talking about right there is called phagocytic lymphohistiocytosis, which is a nice word. And generally, the typical patient that suffers from this is like very young babies that upon their first viral infection gets to this state. And if they don't, if they're not treated with heavy cytostatic treatment and in the long run bone marrow transplantation, they will die. So this is a very, very severe condition. However, in recent years, it's been, and I should also point out that this is due to mutations in single proteins. So it's a little bit different from what we talked about before. I mean, in the previous very interesting talk, we heard about the SNPs, these single nucleotide polymorphisms that we all have. And they're generally in the polymorphic regions of the genes where you have a lot of variation. And I mean, one thing that Nancy didn't point out is that we wouldn't all just look like Nancy if we just had the same SNPs. We would also all be dead, because that's sort of, it's a very important thing that we all have. You know, we have these tweaks of the immune systems and our bodies generally so we can cope with different dangers in different situations. These patients, however, they have mutations in conserved parts of the genes that are important in this process, which renders them very susceptible to this. But we've done, and we and others have found that you don't always have this 100% impairment in the cytotoxicity, but rather it can be a 90 or 80%. And depending on what level of deficiency you have, you will end up with a susceptibility to different disorders. And as we're sort of a diagnostic group, we get patient samples with these kinds of disorders, hundreds of them each year from different clinicians in large parts of the world. And we diagnose them and we identify their mutations in these specific genes. Then we have a pretty robust set of diagnostic assays for these things, but we're then also interested in trying to find other patient groups where you could suspect that there's a sort of milder reduction in these functionalities. And then, so that's something that we're constantly doing. We're trying to find these patient groups. So that's sort of our entry into the ME field. As you know, I mean, it's already been shown that already in the 80s there were studies showing reduced cytotoxicity in NK cells from ME patients. And there's also been nice studies showing that regardless of, if you get a viral infection, it can be sort of many different viral infections still render you a similar susceptibility to end up with ME after half a year or a year. And that, at least with our sort of immunodeficiency, our immunodeficiency view on this, it could be talking in the direction of something going on in the immune system. So our hypothesis into this field was that ME might represent a syndrome where a substantial fraction of the patients have impairments in lymphocyte cytotoxicity, and that this could then be sort of causative. So that was our most basic idea. The second thing would be that the refined diagnostic assay that we're using for other immunodeficiency syndromes, it could be used to detect such impairments. So with that background, we decided to make a pretty, it's not a very big study, it's a pretty small study. It's a total of 50 patients and 50 controls. But what we've been doing all through is that we've been taking all the patient samples and all the controls, and we've been doing all the experiments on everything together, because we know that with the assays that we're using, we have a lot of inter-experimental variability, and that could sort of take away small subtle differences. So, and this is maybe a key point actually that differs from previous, lots of the previous NK work, and that might explain part of the differences that we have. So we're freezing all the cells, and we're thawing them, and we let them just rest overnight, as we say, because they're really stressed. The cells are stressed by the freezing and thawing. And then the next day, we have a decent viability of like over 95% generally of the cells, and we run the assays, and they respond. So what we're looking at is then whole blood cytotoxic lymphocyte counts, cytotoxic lymphocytes then being both T cells and NK cells. That's naturally done then in whole blood the day when the patient comes. But then we're looking at the cytotoxic granule content, so the perforins and the granzymes. We're looking at the capacity to release the cytotoxic granules, and we're looking at NK cell killing ability. And these are the things I'm going to talk about today due to lack of time. We've also been looking at cytokine production, baseline activation status, the frequency of CMV-associated adaptive NK cells, and we've also been looking at some in vitro regulation on cytotoxic lymphocytes by adrenaline. I'll briefly mention that if there's time. We've also been doing some like multivariable analysis to look at very weak, if like small problems in multiple of these factors could contribute to together make like be a difference between the patients and the controls. And I have to say that we haven't been very successful there in the last step. But then we're trying to select variables that separate patients from controls, and then we're retesting. This is a central thing for us. We're retesting our findings in a second independent group of patients. So we have one group, I'll go to the next slide. We have one group of 25 patients and 28 controls from Stockholm, and then we have a second set from Oslo. And I guess part of the explanation that we've been able to achieve this is that my supervisor is actually Norwegian.
that happen to immigration policies?
It's a good question. They're still pretty. I mean, they're still pretty open, I think, to Sweden compared to other countries. But
Scientists don't worry about those.
No, that's true too. That's true too. But as you see here, there's one clear difference between the groups, and that is that the median age is significantly higher in the Stockholm group than in the Oslo group, with a median age of 44 years in the Stockholm group and 30 in the Oslo group. But there's no difference between the controls and the patients in each group, and we've also all the over 50 things that we're looking at, there's not a strong correlation between age and these factors. That shouldn't have anything to do with the results. You also see here that they have a long duration, and when it says infectious trigger, I mean, essentially all of them have infections are involved, but in these cases the infection, there's been a very clear correlation between the onset and the infection, whereas in other cases it might be a more gradual onset case, and that's more so in the Stockholm group, where a few patients have this, whereas in the Oslo group almost all have a very clear-cut infectious start. So over to the results. I have to see them. Okay, I can instead of looking like this, I can understand it. First, we just looked at the whole blood counts on the different cells. And here we don't really see any differences in the whole blood counts, which goes against some reports, but not others. I think some have reported a different frequency in the 56 bright NK cells. So what we're looking at here is the 56 dim and bright NK cells, and we're also looking at the 57 positive and negative CD8 T cells. The 57 positive being the ones that are the main cytotoxic effector cells, and the others are more here for completion, sort of, and they're less cytotoxic, they're naive and different memory subsets with other functions. Then what we've gone on with doing was to look at then Perforin and granzyme A and granzyme B, and especially Perforin has been implicated in a few different publications previously. To our surprise, we didn't really see any differences here in the levels, both when you were looking at group level and if you're looking at more like subgroups. You see that the patient group, which is the black bars, they're pretty dense. They're sort of, most of them are pretty closely together. We do, as you can see, in the 57 negative CD8, so the non-cytotoxic cells, we do see a tendency towards the difference on the group level for granzyme A. Then we did the same thing in the Oslo group, and here the differences are very much smaller, and it's hard to interpret with these kinds of numbers. One thing that I should mention, though, is, and it's a bit hard to jump all the way up there to point at it without the pointer, but the Perforin, on the upper side, you see that anyway, for the Stockholm group, the black dot, you see that there's a black dot in the Perforin graph on the lower side. And that black dot is the same patient in all 3 cell subsets. That's not true for anything else here. All of the other places, it's sort of mixed. And that patient turned out to have a variant of the Perforin gene. So I actually asked Nancy about that, if they had looked at that in their really nice material. So this variant in the gene is one of these SNPs, and it's been previously shown to lead to a slight reduction in the Perforin level. But it's also present in the general population at a frequency of about 1 in 400 people. So it's very hard when you only have one to say anything statistically. So this is a too small study to address this. But that's definitely something that I would be very interested in seeing in one of these bigger genetic screening materials. But apart from that, this data, at least with all the different ways that we've tried to look at it, seems pretty negative, and we were surprised by that. Then we went on to look at cytotoxic granule release, because as I said before, you not only have to have the proteins, but you also have to be able to release them to be able to do this, and to be able to kill other cells. And I mean, making a long story short, as you can appreciate by just looking at each single pair of these, like these ones or something, you see that there's no... Yeah, exactly, it's true. I thought it was okay to just show one example, but we can try. It becomes something else than a normal lecture. You can appreciate that there's not really any differences, and we've been investigating a number of different stimuli. And actually, an important thing to point out during all of this talk is that we can't say with any of this that there's no defect under any circumstances in this process because we haven't looked at everything, right? But we can say that with these patients and with this experimental setup, we don't see any difference when we're looking at these aspects. So I won't go further than that, but that's something that I feel is pretty clear now when I've been doing this back and forth and done it in 2 different cohorts. Then, I mean, one thing that has been also today and previously shown many times is that the NK cell killing is reduced in the patient cells. And so we naturally went on and we did that as well. And then to our surprise, in both groups, we didn't see any difference here. And this is my most controversial slide, naturally. And I'm very happy to discuss this. I don't know, I'll just go through what this slide, what this graph actually shows. So on the x-axis, you have the NK cell to target cell ratio. And for each patient, we're incubating the cells with different concentrations of the NK cells. And actually what we're doing here is that we're having a mix of cells, not only NK cells, but PBMC, so T cells and B cells and other cell types all together. And then we're calculating the percentage of NK cells from a separately performed sort of counting assay that is done in parallel and as long. So it should really reflect the number of NK cells. And then we're looking at the lytic capacity on the other axis. And then these small graphs there and there, they show if you connect all the 6 different concentrations for a single individual and make a line out of them, and you just take those regression coefficients and you put them all together, you can see that there's not really any difference between the patients and controls here. Let's see what I have here. Okay, so what, I mean, in a way, as I said, we've also looked at a number of different factors, but we don't really see anything in any of those other factors either. So there's no point in showing you a lot of negative data, and I think that this is sort of the most central slide, and I'm very happy to discuss it. But I just want to mention sort of at least my theories about this a little bit. And as I said, the one major difference between our setup and other setups is that we've been freezing the cells, thawing them, and also resting them overnight, because this is sort of the convention in the immunodeficiency diagnostic field. This is what people normally do. So there are 2 problems or 2 methodological differences that this leads to, as I see it. One thing that was pointed out the other day was that naturally, if a certain percentage of sick cells die during this process, and that only happens in the patient samples, yeah, then naturally we will have a problem. There are, however, a few things that speak against that. And that one thing is that we don't see reduced NK cell frequency compared to any other cell types. So then it must be that all different cell types that we're having together here are dying at the same frequency. And we also, already there, that using Occam's razor, that's a very complex sort of explanation for this. The other possibility, as I see it, is that what we're taking away here is all the influence of all the serum factors. So maybe there's actually an important interaction between soluble factors and the NK cells that render the cells functionally incompetent in vivo, or if you take them straight out of the blood and look at them, that's then when you just isolate the cells and you look at the cells, you don't see this difference. And at least to me, that might indicate that this is not. I mean, I don't really want to say this so straight because it's maybe too strong, but in a way it might not indicate that it's the NK cell problem that's causative, if you see what I mean. If you create, for some reason, you create a dysfunctional environment in the blood that leads to a deficiency in the NK cells. Okay, thank you. That can mean, I have 5 minutes left, that's good to know. If you have this, then naturally that will lead to a lot of consequences. You might be much worse off responding to other infections, but it might still be that there's some other factor that led to this in the first place. That's at least a possible interpretation. So we didn't have any good systems to check this. I've realized afterwards that what we should have done to test that hypothesis would be to just take patient serum and control serum and incubate the cells together with them and see what happens. And we haven't done that, so that's sad but true. But I encourage anyone to do that experiment. I mean, it's a good one. But what we have done is that we've, maybe I shouldn't show it yet. So one thing that we know is very important for inhibiting NK cell function is adrenaline. There are naturally many other factors, like cortisol will also have lots of effects, but one thing that we know is an acute effect is if you're running or if you're standing on stage like me talking, you get an immediate increase of cytotoxic lymphocytes in your blood, and these cells are, at least short-term, clearly functionally inhibited. This has been shown for 30 years, and I also have a publication on that in a slightly different setting. This means that if ME patients, when they're going to the doctor, have a higher baseline activation of their adrenaline system, or if just the fact that when they're going to the doctor, I mean, we know that, right? I've been talking to quite some patients that say that just going to the doctor is a major effort that will lead to them being maybe bedridden for a week. And I mean, naturally, it's not unlikely that they will then respond with a strong catecholinergic response just during this thing. And that might render the cells hypofunctional during a period of time. And then what we tried to do was then to mimic that to see if there was any difference in the response to adrenaline, just in vitro. And then we incubated NK cells together with adrenaline at different concentrations for 15 minutes, and then we added a strong stimulus, and then we see how they reacted. And as you see, there's not any major difference in the response between the groups, which sort of speaks in the same direction, not towards an internal difference in or an internal defect in the NK cells, rather that they respond normally. But if you're already here, if you already have a high concentration of adrenaline in the blood, then that will render a difference in vivo. But what we did see, as you can see here for the lowest one that I'm pointing at here, is that there is a small difference where the black curve is slightly higher than the gray curve. And I'm showing the range here, so that's why the arrow bars are so wide. And that's actually a significant difference between the groups. And at least that's a complete speculation, but you could imagine that if you're having a high catecholinergic stimuli all of the time, you will probably downregulate your adrenaline receptors as a response to that. And that would render you, in an assay such as this one, less responsive to adrenaline. And that's exactly what we're seeing. So the cells can still respond, although they're under this stimuli. And that's also been, similar things have been shown by previous authors in slightly different settings. So with that, I would like just to say that, I mean, as in this little study with a different focus and a different, it's different in some ways. We don't really see any difference in lymphocyte cytotoxicity. This might be due to isolation of cells or other issues. But I would then like to ask the controversial question, if there is such a dependency on the experimental setting, is this useful as a biomarker? That's a very controversial question, actually. It depends on what you're going to use the biomarker for. Maybe it is anyway, because as has been pointed out before, I mean, NK cells it's reasonably easy to assess them. But I mean, if you could identify the serum factor that inhibits them instead, maybe it's easier to assess that directly. I'm not sure. And with that, I would like to just acknowledge all the people that have been involved, mainly our clinical collaborators, both Elin Bollestrand and Indri Jungar, who are both here in the audience, and all the patients and controls, naturally, that have contributed greatly to this. Thank you so much.
Time for a couple of questions.
First of all,
Hi.
let me say that the way we measure this function in patients, which we do every day as a clinical test, is we use whole blood. We don't separate the lymphocytes.
Exactly.
They are in their natural milieu.
Yeah, I completely agree.
I would invite you to come into our laboratory and spend a couple of weeks, and we'll show you how we do it.
No, but I have to say that I agree. I mean, and I think this is a very important thing. And this is something, we're writing a manuscript on this, and this is something that we're really saying, that we know that you've been doing this in whole blood, and I mean, that's a very valid thing, and we're also establishing these kinds of assays. But it depends on the question, and also the answer can sort of maybe lead to different interpretations of the biology behind this.
I mean, we can also show that this property of having low activity from the whole blood assay is associated with ME/CFS.
Absolutely, yeah.
And if it's also an important marker to be trying when you do a clinical trial or anything like that, you can measure this function, but you have to do it in a certain way or you won't get the same result.
And I mean, I think this is a very, very. I tried to say that I agree with you. Really, I mean, I'm sure this depends a lot on the experimental setting. But what I'm thinking is that if this defect that you see when you look at whole blood is due to some soluble factor that we can measure, for many labs it's very complicated to set up a whole blood functional assay. And if we can instead identify the factor. Yeah, okay, it's easier, yeah, but still, you will need a sort of cell biology lab. If you would instead be able to measure some other factor, it could be done in a clinical diagnostic setting, maybe. Because I know that the whole blood functional assays are pretty far away from the standard diagnostics. That's what we experience in our field, anyway. But I agree. I mean, this is an important point. Other questions?
I just ask if Dr. Unger is available, if she would comment on her work in Perforin, if I'm pronouncing it correctly, and how that might fit into this.
Is Dr. Unger in the audience?
Okay, but I think that we've already had, part of that discussion was already up before, right? That this is a difference between different labs already before. Some have seen a difference in Perforin mRNA expressions, whereas others haven't, if I'm not incorrect. So there's already a discrepancy there between different laboratories. So, I mean, also what we've been discussing a lot during the last days is that there might very well be that if you're looking in one center, you might identify such a difference. But when you go to another center, due to the population or due to some small diagnostic differences, you end up with a slightly different population and there you don't see it. So, I mean, these things are perfectly possible. That's why we set this study up with two separate independent cohorts. Yes? Do we have a... Yeah, you can yell. Great.
I'm curious what kind of a serum factor you might think would be responsible, because we're also seeing some evidence, and Ron's going to present this later, that there could be something in the patient's serum that's either there or lacking that's causing a specific phenotype. So what sort of a factor would lead to that?
I mean, as I sort of showed, already such a factor as adrenaline could be responsible for this effect. I mean, there are quite some studies showing that the HPA axis is sort of imbalanced in these patients, and we know that that has long-term effects on the cellular phenotype and function. And also, it could be. I mean, we will hear more about lactate later. It could be that due to, I mean, it could be secondary effects, that you have some factor that like lactate, that leads to a stress response from the body that in turn affects the NK cells. But these cells are extremely intrinsically regulated by all kinds of different factors. And I mean, they express receptors for dopamine, serotonin. They express so many different receptors and are regulated by all subtle differences that you can imagine, sort of. So I can't denote specifically what one.
Thank you very, very
yeah,
much,
okay.
Jakob.